Hey everyone, in my last video on James' tour, we got some eye-opening perspective into how he conducts his research, and today we will see more of that as his colleagues are coming out one by one to blow the whistle on his fraudulence. But we have more of his apologetics to cut through first, so let's see what he's been up to, shall we? For some quick context, you may recall the part of our debate where I brought up systems chemistry. This is the field that studies how systems of molecules interact to create new functions and emergent properties. The exciting part about this field for origin of life research is that we can observe molecules exhibiting Darwinian principles and evolving according to the resources available to them, thereby offering a bridge between chemical systems and biological systems. I've explained a lot of this research in prior content, and it really pisses off creationists because it shines a bright light on how inanimate molecules can have slowly formed the first living organism billions of years ago. For this reason, James does nothing but pretend it doesn't exist and deny the results of literally any experiment in the field. In the debate, I brought up the work of Gerald Joyce, specifically self-replicating ribozymes, and he just said, "Nuh-uh," thereby accusing Joyce of reporting fraudulent data.
Do you agree that this research shows replicators that evolve by natural selection? Because it does.
No.
Do you?
No.
Oh, okay. So Gerald
No.
Joyce is a fraud. Fully replicated.
It is not fully replicated. No way. It is not.
20-fold amplification.
They take a long bit and they duplicate this much of it.
James, how would it retain
But
catalytic activity?
even if they could,
No!
Yes, James' Show Me the Data tour apparently doesn't like data that proves him wrong. I probably don't need to explain how 20-fold amplification is physically impossible if molecules can't undergo even a single round of replication, but we will return to this chemistry a bit later. Since then, James continues to spew hot garbage whenever pressed about this field, refusing to engage with it in any way whatsoever.
What is the systems chemistry, and why do detractors say you need this to understand origin of life? It's a smokescreen. You have to understand every one of these components, and systems chemistry makes it much harder for them. They dig their own grave when they say you have to understand systems chemistry.
It's difficult to articulate how pathetic this is. I think James hopes that if he hides under a pile of coats and shuts his eyes, systems chemistry will just disappear and he won't have to learn about it. And he clearly wants to program his followers to exhibit the same Pavlovian response and recoil at the mere mention of this field, since it's probably the single most important area for abiogenesis. But denial isn't a strong enough defense mechanism for James. He wants to share this delusion with others. So for his latest shenanigans, he enlisted someone even more clueless than him, a creationist named Rob Stadler. I heard of Stadler back when I did my very first video on James, because this guy tried to debunk it. Almost nobody watched, so I ignored it, but now that he's back in the mix, let's go ahead and tear apart the old video and the new one. Here's the first one from 2020. Must watch. Reality check. Oh no, I'm getting educated. This should be good. But wait, who is this Stadler guy anyway? As it turns out, he makes medical devices, so he has zero qualifications in this area, and of course he wrote some crappy books, one on origin of life and one on evolution. What they didn't teach you in school, obviously referring to his science denial and blind faith, since those have no place in any science class. For the one on origin of life, given that Rob has no background in the relevant science, he had to recruit a co-author to help with the technical stuff, and that's creationist Laura Tan, a disgraced former molecular biologist. She used to be a professor at the University of Missouri, but she was fired for low productivity, as is indicated by her H-index of 14 and very low citation count. Additionally, a good chunk of her very few publications were just entries in a creationist propaganda journal called Answers Research Journal, which outright states that its purpose is to highlight research on the recent creation and global flood within a biblical framework. Her firing likely also had to do with her abysmal teaching, given that on Rate My Professor, almost all of her reviews are ones, with students saying that she teaches creationism during lectures. What a perfect match. Anyway, let's get to the video.
The trick is, and the trouble is, that when Dave says it is clear that Jim Torr's, religious views cloud his capacity to interpret research on this topic. What Dave is basically saying there is that only atheists are qualified to address the science behind the origin of life.
No, genius, I am not saying that only atheists can discuss this science. There are plenty of religious scientists who do not allow their religion to influence their view of science. Furthermore, you don't have to be an atheist to accept a naturalistic explanation for the origin of life. Plenty of people believe in God and accept science. So this is a double straw man. I am saying that James specifically lies about this field because of his religion. He can't help but start with this persecution complex because it's all creationists have. He can whine all he wants. James does nothing but lie about this field, which I have proven conclusively over the past several years, and he does so because of his religion. Anyone who can't see that is blind or brainwashed.
In order to be an atheist, in order to be a rational atheist, you have to have a natural explanation for the origin of life.
No, you don't, at all. We used to not know what lightning is, or volcanoes, or disease, or literally anything else. Some people believed these were all deities, some didn't, and we slowly figured out what all of those things are. You don't have to be omniscient to be an atheist. Sorry.
If our judgment was truly clouded by our belief, then how come the amount of Nobel Prizes are disproportionately in favor of Christians over atheists? If our judgment was clouded, we would not be dominating in that area.
He's already a broken record. Again, scientists can be religious. They just don't use religion to do science. The realms are separate. They didn't win accolades for proving God exists. They got them for doing science, which has nothing to do with God.
Rob, that even if modern laboratory experiments could create life from non-life, we would have no confidence that the conditions of the laboratory experiments were consistent with those of the early Earth. So I just wanted to point that out because I really liked that in your book. So
Yeah, if you could do it in a lab today, it certainly would be encouraging. It would be very interesting, but I wouldn't say that that's proof that that is relevant to what actually happened.
And of course, he has literally no idea what origin of life researchers do. They aren't specifically trying to create life as a primary goal. They're examining how systems of molecules can self-organize and self-replicate under certain conditions, thereby elucidating the pathways by which life arose, which will never be even remotely comprehensible to this roster of loser apologists who will never lift a finger to learn any science.
Ribozymes can even perform self-replication. That's right. Already at this simple level, we are introducing the possibility of self-replication.
Okay, hold it there. So Dave is making a claim there that molecules, especially ribozymes, which are RNA molecules, are able to self-replicate. He said it in two places. I find this very disturbing because the fact is that no one has ever seen a true self-replicating molecule. No one ever.
20 minutes in, he finally tries to actually discuss some science. He doesn't like that I mentioned self-replicating ribozymes and claims this has never been done. Incidentally, this is what James also says, no matter how many papers I show him where precisely this has been done. When apologists preach to their congregation, they can lie as much as they want, but when shown literature that proves them wrong, they have to resort to pretending that the research is somehow either fraudulent or irrelevant. The title is misleading. Dave can only read the titles. No, that's just a desperate attempt to invalidate an entire field of science without actually engaging with it. I've discussed a number of these papers in detail. Self-replicating ribozymes via ligation, and more recently, nearly fully self-replicating via polymerase activity. You can refer to my conversation with James Atwater from my video fact-checking Jim's debate performance for a summary of precisely this research, which echoes everything I've said about it.
Undergoing self-replication with exponential growth. So there you have it. It's done. But when you actually read the paper, once again, it takes a very, very charitable definition of self-replication.
Now begins the slander of Gerald Joyce. He has to in some way address the experiments that have demonstrated autocatalysis, so quite predictably he jumps to whining about how this RNA strand was not produced prebiotically.
So in the yellow, you see a string of 66 ribonucleotides with the codes. That is a molecule there that was designed by human intellect and passed through, a sort of artificial selection process, a laboratory evolution process, if you will, with human intellect pulling out the winning molecules. And so they arrived at this molecule because it has the property we're going to talk about. But that molecule in yellow is by no means something that you could find in a prebiotic world, in a place before life existed.
No shit, Sherlock. That's not what the research is about. It's about autocatalysis, not prebiotic synthesis. Apologists pretend that any experiment in abiogenesis has to go from diatomic molecules and lightning to a living cell in one pot overnight, or it isn't valid.
Even looking at just the chirality of this molecule, this is a homochiral molecule. You took all the mass of the universe and randomly had it be 66 ribonucleotide strings, you still wouldn't be able to find one that was this particular molecule because it's that rare, that hard to come by.
Joyce is examining the autocatalytic properties of certain RNA molecules and nothing else. So when Rob whines about homochirality, he sounds like an idiot. When he whines about all the mass in the universe and probability, he sounds like an idiot. This is research about what RNA can do. Anyway, to avoid acknowledging the validity of this research, he complains about the ligation mechanism and how the ribozyme is joining two halves together rather than individual nucleotides.
So when the yellow molecule brings the green and the blue in close proximity, that PPP is very eager to bond, and it creates a bond between the green and the blue. And then there it is, you have a copy of the yellow. So that is considered by Gerald Joyce to be self-replication.
Of course, that's why he is pretending no other research exists, rather than discussing more recent work where autocatalysis occurs via polymerase activity. It's like pointing at the Wright brothers' prototype and saying how ludicrous it would be to claim that planes could carry hundreds of people across the country.
Okay, so here we're trying to build a protein from individual amino acids. And Dave is kind of hand-waving his way through it to kind of make it sound easy. But you have a problem in that these amino acids, when they join together into a polypeptide, there are multiple ways they can join each other, and only the alpha peptide bond is what we find in life. But when that alpha peptide bond forms, it kicks out a water molecule. So once again, if this reaction happens in water, it's going to be driven backward. It's driven the other way.
You are likely starting to see how Rob graduated from the James Tour School of Science Denial. Remember when James whined about how you can possibly get peptide formation in water, and then I showed him dozens of examples of aqueous prebiotic peptide formation, and then he just pretended I didn't for over a year and wrote Clueless with chalk? Rob's taking the same approach here. He goes on to bring up one paper, lie about what it says, and pretend it's the only paper that demonstrates prebiotic peptide chemistry. This is why everybody makes fun of apologists, especially when they whine about atheist bias. Nobody projects harder than them.
Each ribonucleotide in RNA has 4 chiral locations, or 4 stereocenters, and that means there are 16 possible chiral forms for every ribonucleotide. And I've never seen anyone try to propose how a natural process could select out one out of the 16 possible forms and choose that. Because if you have a solution with all 16 forms in it, the chosen ones that you like will never find each other, will never bond to each other and make a perfectly homochiral molecule. The probability is just completely against you.
The pageantry continues. When creationists complain about homochirality, what they are saying is that they can't figure out how molecules of a particular configuration polymerized, therefore God must have linked them together magically one by one. This is the God of the gaps. They don't stop and think about what they could be missing, like thermodynamic reasons that certain forms would predominate over others, or kinetic reasons why certain forms would polymerize faster, or degrade more slowly, or crystallization processes that would result in enantioenrichment. They want it to be that God wished them together with magic, so they deny science they don't want to exist in order to reach that conclusion. That's the bias. In reality, I've gone over so much research in this area, much of which was from Donna Blackman. Here's another paper by her which talks about glyceraldehyde as the key chiral building block for sugars, and that steps are diastereoselective. The explanations involve some of the stuff regarding eutectics we talked about in previous videos. It's complicated and really not worth explaining at the moment. The point is simply that mountains of literature exist addressing these concepts. Rob is just a delusional moron who reads absolutely none of it and then writes books pretending to know what he's talking about. Typical creationist.
We know through directly observed genetic evidence we have, such as the long-term evolution experiment of Richard Lenski and many others like it, we know that living organisms will over time simplify themselves to be the simplest that they can be and still survive. So what I'm saying is that these single cell organisms are trying to simplify themselves, and they've been doing that as long as life has been around. And so if you want a protocell, just look at the simple life that we have found living on our planet right now. That's as simple as it's going to get. It can't get simpler. It's tried. It's already tried. That single cell organism contains 468 genes and about 580,000 base pairs of DNA. So 580,000 letters of code in the DNA. And that is a very simple living organism.
This is probably the dumbest thing he says in the entire video. This is like pretending that a modern building is the simplest dwelling that can possibly exist, because if you take away the chimney or some central steel beam, it will fall apart. What about this hut made of twigs? No chimney there. No, Rob, extant life is not as simple as it gets. That's totally idiotic. It's the product of billions of years of evolution and the acquisition of novel metabolic processes and morphology. Just because certain functions can be lost given the lack of certain stimuli, like he complains about with the LTEE, it does not negate the billions of years of evolution of prokaryotic organisms. It's like cutting out a person's heart, watching them die, and saying, "See, all life needs a heart." No, humans need hearts. Life existed before hearts did, but things with hearts that are alive today can't live without them. Here's another analogy. When examining ecology, we can come across a complex ecosystem with a rich network of interdependence. There is total reliance for survival among the many species of plants, fungi, animals, and microorganisms. If you remove any singular species, the whole ecosystem could collapse. So how did it get that way? The whole thing must have been created exactly as it is today, right? No. Ecosystems respond to the stimuli that are present, minor perturbations, slowly shifting inputs, and interdependence can emerge gradually. Tiny changes over long periods of time allow for a gradual response that produces a more robust system. A sudden, dramatic change, like deleting an entire population of organisms, has a much bigger impact. That we can't start stripping away components of the ecosystem doesn't mean that simpler ecosystems can't exist, or that this specific ecosystem wasn't at one point much simpler. Back to the point, extant life can't be arbitrarily made more simple, even bacteria. Life in general can be. So whining about genes when the first protocell didn't have any DNA at all just makes him look like a moron.
Common sense has no place in science or in an assessment of the feasibility of biological processes.
Okay.
Reasoning.
Hold it there. So Dave said common sense has no place in science or in an assessment of the feasibility of biological processes. So I guess we should at least give Dave credit for practicing what he preaches. He's asking you to put aside your common sense, suppress your thinking, your intuition, and just close your eyes and believe. That's what he wants.
Love it. And we wrap this video up with Rob making a complete fool of himself by demonstrating that he has no clue how science works. He doesn't like this quote from my video that slams people who think science should be common sense. Anyone who has ever done science knows that common sense does not guide scientific inquiry. Science is often, if not usually, unintuitive. There is nothing whatsoever about chemistry that is common sense. We probed the molecular world and ascertained its laws through empiricism, which are completely and utterly removed from the everyday experience that forged our common sense. What goes up must come down is common sense. That conjugated pi systems undergo cycloaddition reactions is not common sense. That electricity and magnetism constitute orthogonal oscillating fields is not common sense. That stars are powered by nuclear fusion is not common sense. That heredity is determined by a double-stranded polynucleotide is not common sense. That the human body can attack itself via autoimmune disorders is not common sense. That the Earth goes around the sun instead of the sun going around the Earth as it appears in our subjective experience is not common sense. Pretty much nothing ascertained by science after around the 12th century is common sense. It is arrived at through diligent empirical analysis. There can be no better demonstration that Rob is profoundly clueless about what science is and how it is done. So as you can see, he needed over an hour to discuss maybe 4 concrete scientific points, and he fumbled all of them. Now that you're aware of the level of intellect we are dealing with, let's watch Rob and James make fools of themselves together in the latest video.
It's rare that somebody will come on my podcast anymore and talk about origin of life issues with me. I've asked many, many experts to do that, and very few people will do that.
Yeah, and astronomers typically don't go on flat Earth channels to explain science to them either. It's kind of pointless. After that, he whines about the Harvard event a whole lot, spewing all the same garbage we saw in the last video.
This whole idea that I was shouting, I was hurting people's ears, but the only way that they could get Lee to come is to assure him that I wouldn't interject. Well, Jim says I'm a lousy chemist. I never said that he's a lousy chemist.
He again pretends it was a debate when it wasn't. He again whines about being accused of shouting. He again pretends he was told not to speak during the dinner portion. He again pretends he didn't slander Lee after literally calling his research garbage during his opening statement. Boy, is he desperate to shake off the severe humiliation he experienced over there. Next, he regurgitates some tired old claims about the chemistry.
You got all sorts of structures that are not polypeptides at all. What he made was a barrage of oligopeptides, totally disordered, and the average was like a 12-mer or 14-mer, tiny little things. That's all he could get to make without doing other synthetic techniques using prebiotic chemistry. And whenever he had the side chains there, it had to have had branching. He said he had linear ones. There was zero experimental evidence that he had any linear ones in there.
Yes, again with the peptides. I refuse to explain for the 100th time how peptides can form in water. James has not bothered to touch any of the papers I've shown him and just lies about them, only to shift the goalposts to whine about reactive side chains and his ultra-specific decay linkage. And of course, he continues to ignore everything I've said about how this is a straw man, since there is no reason to believe this specific linkage had to be present for life to arise. And of course, he continues to ignore the Pounder paper that shows peptide bond formation with no interference from reactive side chains, which even the dishonest duo over on the Christian Apologist channel admitted solves the problem. But that's James Tour for you. He sets his own idiotic rules and then gets angry when people who actually understand this field don't take him seriously. Just for fun, here's a paper which deals with self-replicating peptides. Yes, it's not just RNA that can do this. This paper shows that if an amide bond is formed with the terminal side chain, or if the amino acid racemizes, the peptide that forms loses much of its ability to replicate. In other words, there is an automatic selection mechanism for linear homochiral peptides. Seems pretty relevant to the origin of prebiological selection, huh? But of course to James, it all has to be nonsense and garbage, because reasons. Here's yet another paper from all the way back in 2002, which shows peptide self-replication approaching exponential growth. Here's the data, and the author specifies that the efficiency is very close to naturally occurring enzymes. James and pals don't look for this stuff because they don't want it to exist, and they refuse to acknowledge any of it no matter how many times I show it to them.
Molecules have never been known to move toward life, ever. They don't know to move toward life. They have no brain. They don't know what target they're going toward, and but they try to force it, and it's just not working.
More of Jim's favorite mantra: molecules never, ever, ever move towards life. So it had to be God, but not the God of the gaps somehow. Well, James, we certainly have shown molecules to move towards increasing complexity and systems that more closely resemble elements of evolving life forms with no guidance other than differing replication rates and natural selection. Lots of papers describe this. How does that happen, Jimbo? Care to explain it to me? Is God sitting in that flask pushing the brainless molecules around?
I felt like there was a little bit of a self-conflicted message coming from you, because in one point you say that prebiotic reaction chemistry fights against a path that moves toward life. In other words, saying that what we've learned already is telling us that it opposes the formation of life. But then later in the presentation, you said, this is like 2 hours and 15 minutes into the video, when, not if, but when someday we do figure out how life got started. So which one is it?
I'm glad you asked me this.
Rob finally opens his mouth to pose a mild challenge. James always says that molecules never move toward life spontaneously, which implies a designer and is exactly what Rob wants to hear. On the other hand, James has said, although totally disingenuously, that eventually we may find out how life began, something Rob does not like. Isn't this a clear contradiction? You'd better believe it, and James can't wait to clear it up.
As scientists, we cannot say what we will never be able to do unless we are violating a strict law, say, of thermodynamics. So, for example, based on thermodynamics, we will not be able to make a perpetual motion machine, something that has zero input yet keeps on running. So there's no law that says life cannot form based on a materialistic model. So how can I say that we will never be able to do it? I can't say that. All I am saying is that there are many discoveries that we're going to have to make before that will ever happen. Many more of these are going to have to be discovered before we can ever see molecules projecting toward life, in my opinion. So I'm entirely consistent. We're nowhere close. The target is moving away from us faster than we are moving toward it, because we learn about the more complexity in the cell.
Since he is in good creationist company and not real scientists at Harvard, he admits that it's creationism all the way. But Rob keeps pressing, and things get even more ridiculous.
Then we learned enough to be confident and say, you know what, it's impossible. We literally cannot possibly have a perpetual motion machine. And so I'm wondering if the next 100 years of science and learning about natural laws through that process, in your mind, is it possible that that would lead us to say, we know enough to know that natural processes could not have started a living thing? Can we ever get to that?
Absolutely. If that's where the science suggests, if we can derive laws that suggest that we'll never be able to solve this thing, boy, I'll buy into it.
Yes, can we ever get to the point where science proves that natural mechanisms alone can't explain the origin of life? And James agrees. These two idiots who call themselves scientists are saying that science will demonstrate miracles, and we are just around the corner from it. At one point, Rob even tries to pull out some popper to say that abiogenesis has no element of falsification, so it's not a scientific theory.
To me, if they're doing real science, then in their mind, it must be possible for what they're working on to be something that could be falsified. Okay? And what I would ask is, for them, what kind of evidence, hypothetically, what kind of evidence could you encounter someday that could convince you that life could not have started on its own? Can you answer that? Is there any hypothetical evidence out there that you would find that could convince you that life could not have started on its own? And if they can't answer that, if they can't even imagine finding some evidence that could convince them of that, that tells me that they're actually not following the evidence. The evidence is irrelevant.
More of Rob showing everyone that he doesn't even know what science is. The basic postulate of science is that physical phenomena follow universal laws that we can discover and use to make predictions. The origin of life by natural means is not a theory, it is the postulate itself. Specific models that seek to explain the origin of life, RNA world, metabolism first. Those are the theories, not the mere concept of life arising. A scientific theory has to be falsifiable, at least according to Popper, but the basic postulate does not. Science is methodologically materialistic, and supernatural events are never considered, otherwise science would collapse. Rob is blissfully unaware of the philosophical basis of science, totally incapable of seeing how his mind has been poisoned by blind faith. Of course, James is in the same boat, so he plays along, talking about perpetual motion machines, as though we will somehow figure out how abiogenesis violates the laws of thermodynamics, even though we've gone over piles of research which show how it's specifically prompted by thermodynamics. Besides, we have actually discovered a perpetual motion machine. It's James never shutting up about this topic, no matter how many times he's humiliated.
And what the community has to now do is pull the primordial soup model out of education, out of textbooks, from the 8th grade all the way through advanced university. It has to come out because nobody can give us an example of how molecules can come together to form a cell, those cells then taking on life. The primordial soup model has got to go away, and they have to begin to concede. We have no idea yet how this could have happened.
And he launches right back into his nonsense about the primordial soup, which again is just a phrase that was coined a century ago to describe life arising from molecules in water before we had any clue what those molecules were and what they did. If we were to describe this model with modern concepts, it would basically just be systems chemistry. We will come back to this a bit later, but James is explicitly saying that this shouldn't be in books, so he is saying it is impossible for molecules to form life, which leaves only his god of the gaps he pretends he doesn't worship. And I won't bother torturing you with yet another clip of him whining about the target moving farther away, CISS crap, interactomes, and all of his other favorite irrelevant buzzwords.
And there's another problem with RNA is that RNA is very unstable. As I talked about in the lecture, is that if you have a 600 mer, which is a very short RNA, by the way, if 600 mer, that would only survive for about 4 hours in water, generously 4 hours in water, which is not very long from a chemical or prebiotic chemistry perspective.
Now we get to the RNA world. James informs us that a 600 mer polynucleotide would have a 4-hour half-life. Where did he get that number? Where else? He made it up. Here's some real science on the matter. At pH 7 and room temperature, a 1006 nucleotide strand has a half-life of 10 years. If the RNA is a duplex, it is stabilized dramatically, and replicating RNA would indeed spend most of its time as double-stranded RNA because strand dissociation is the slow step of the process. Other macromolecules could bind the strand and make it even more stable. Sorry, James, you have some reading to do. He also pretends that RNA has to have 600 nucleotides to provide catalytic function. That's another whoopsie. There's no firm lower limit to this ability.
He's talking about an RNA molecule with 188 nucleotides.
Then Rob decides to pivot to a self-replicating ribozyme with 188 units. Just like before, he botches everything, ridicules the paper he didn't understand, and claims that this represents the state of the art, so the field has failed. We will point out his many errors in a moment. First, let's get some context. Here's an amazing paper from Gerald Joyce and Jack Shostak, which examines the birth of protocells, so it's more comprehensive than mere replicators, but it talks a lot about replicators as well. As we already know, one of the prime candidates for early replicators are simple but functional RNA structures, and non-enzymatic template-directed polymerization of activated nucleotides has been the subject of many studies, as it is widely regarded as a reasonable proposal for the emergence of replicating informational systems at the origin of life. To put it a bit more simply, when a functional strand of RNA is fed enough activated nucleotides, a complementary strand can arise due to simple base pairing. So U will pair with A, G with C. You remember high school biology. At the end of this process, the strands will dissociate, hopefully quickly enough that further replication can take place, whereby the new complement will produce another copy of the original strand, and so forth. This happens on its own, no enzymes necessary, even if slower and with lower specificity than what we see in biological systems with enzymes. So this is autocatalytic behavior, which is very likely involved with the origin of life. Because of this, apologists can do nothing but point out issues with efficiency and specificity and pretend they are bigger problems than they are, which they achieve by lying. First, there is the low fidelity. The pairing energy of GC is relatively low, so many of the mistakes involve forming a GU pair instead. These are referred to as wobble pairs. Forming one or more wobble pairs in sequence slows down the elongation of the strand, resulting in low yield, where yield refers to synthesis of a fully matched full-length strand. When we look at Joyce's work, we see a 2% yield for his hammerhead ribozyme after 1 hour using his new polymerase, and 28% after 72 hours. So at 1 hour, 98% of the strands are shorter than full length, though they can be brought to full length with more time. They will simply contain one or more mutations. The 28% represents all full-length copies, even if not faithful copies, with an average fidelity of 83 to 92%.
And in living things, we know that the sloppiest living thing that can stay alive to replicate its own DNA, it makes a mistake 1 out of 33 million nucleotides. So that's the world record of sloppiest replication in the living world. 1 mistake out of 33 million. What they have produced is 1 mistake out of 12. So they're 1,000,000 times off what we know a living thing needs to survive, okay?
Rob laughs at the yield and fidelity, comparing them to modern cells, which have very close to 100% yield and fidelity. They will always compare everything to modern cells because that's their script. Everything they ever talk about always comes back to this dishonest comparison. But let's continue describing reality. The logical question would be, what length of RNA is required and what is the maximum error rate allowed in polymerization such that the strand is selected for and effectively replicates? According to Joyce and Shostak, a 25 nucleotide strand could have done this. For a ribozyme of this length, an error rate of around 2% is tolerated, so perfect replication is not necessary. Even for longer strands, with a 100 mer, there can be an error rate of around 1%. For a 200 mer, around 0.5%. The perfection of modern life is not needed, and in fact higher mutation rates at the origin of life were likely crucial in more rapidly finding useful sequences. And there's more. As a solution to finding this low error rate without enzymes, they've suggested that early RNA may have used 2-thiouracil instead of uracil. Sulfur is ubiquitous in nature and plays a big role in protein chemistry. It has been shown that thiouracil has a stronger pairing energy with A than uracil, and with this modification, error rates around 1 to 2% have been achieved. This is just one example of a totally viable solution that has been proposed that dishonest creationists would never touch with a 10-foot pole. But despite their willful ignorance, we are indeed making steady progress in the direction of prebiotically plausible, sustained replication, and we are nowhere near clueless, no matter how many times James shouts as much. Now, with all of that for context, let's hear a bit more from these two.
So 188 nucleotides is quite complex for a ribozyme. And the chance of something like that randomly producing itself in a prebiotic world is far less than the chance of finding one specific atom somewhere in the universe.
Another entry in the classic creationist script, whining about the probability of that specific sequence forming, instead of focusing on the principles the research demonstrates, which can be applied to countless other sequences.
Even if they had this in 100% yield, then what? Just to have an RNA is not life.
Wow, good thing that James the super genius is here to tell us that RNA is not life. Yeah, we are talking about systems of molecules that produced life over time.
Nobody solved the mass transfer problem. So this is where how much material do you start with? So say I go to the laboratory and I start with a kilogram of material, that's 2.2 pounds of material. I take it on a number of steps. Say I take it on 10 steps, but now I've run out of material because I've tried many things, because the yield, this is the problem that Rob was talking about, the yield may have only been 1%. So I take that 1% and I bring it on. The yield of that next reaction may have only been 1%. So it's 0.01 times 0.01. So you take your kilogram multiplied times 0.01, and you take that number and multiply it again times 0.01, and so you run out of starting material. So what we do in the lab is we go back and we start making more, now using the chemistry that we've worked out. But nature doesn't have that advantage because nature never kept a laboratory notebook. So when it runs out of material, it's over. It's done. There's no going back. That's the mass transfer problem.
Again with the mass transfer problem, which is James, the laboratory synthetic chemist, proving to everyone that he can only understand chemistry one narrow way. Start with X grams of starting material and make one modification at a time until you reach your target. If you run out of stuff, you need to start again. Newsflash, that's not how nature does chemistry. Nature works with complex mixtures and carries out thousands of reactions at the same time, while all the molecules interact with each other and influence the rates and selectivities of all ongoing processes. Nature doesn't run out of material because of some imaginary instability. Reactions are reversible, non-replicating polymeric structures are degraded, and their building blocks can be reused within the network to amplify what is successfully replicating. This is probably the best example of how someone like James, who pretends to have so much relevant experience, specifically does not understand this field. This isn't James just lying, he genuinely does not get it. Nature doesn't need a lab notebook. Get a new script.
Nobody's ever been able to make the polymers, be able to take the small molecules and polymerize them into the larger molecules that make us up. The polypeptides, the polysaccharides, the polynucleotides. Nobody's been able to make those.
James truly has no shame. All of this has been demonstrated right to his dumb face, and he just continues to pretend it doesn't exist. Peptides and oligonucleotides have been made by prebiotic means dozens of different ways, and polysaccharides were made by enzymes.
These compounds, they say, well, there's been all this time. No, there's not been time. It's because as soon as you make one of these, as soon as you make an RNA, it has 4 hours to survive. If it's a 600 more, 4 hours to survive.
Again with this 4 hours for RNA. This is so easy to disprove it's ridiculous. Apart from the research we discussed, just look at studies testing for the presence of mRNA strands injected from COVID vaccines. Significant quantities are still present weeks after the injection, and that's with molecular machinery actively trying to degrade it. RNA does not degrade as rapidly as James pretends it does, period.
And then there's the whole protein folding problem. So it's not just that you had to have the right sequence of amino acids. Now that you have this, now that protein has to fold up into a three-dimensional structure. Because what happens is if you have a 100-mer, just 100 units long, it would take 10 to the 95 possible permutations. If something is over 10 to the 40th, there's not enough time in our universe. So 10 to the 95 isn't just two universes. It's a trillion universes of time.
Now we get to protein folding, Levinthal's paradox. They're saying that if proteins had to sample the entire conformational space to fold, it would take them longer than the age of the universe to find the most stable conformation. But they do fold smoothly and quickly all the time, so how do they do that? Of course, God is sitting there folding them all by hand. Hey brainiacs, the folding isn't random, it's guided by the amino acid sequence and the maximization of electrostatic interactions. All the potential folding patterns are not equally stable, not by a long shot. It's simple thermodynamics. It will fold in such a way so as to make as many electrostatic interactions as possible, thereby bringing the system to the lowest energy possible. That's why proteins have stable three-dimensional structures. The so-called paradox is actually about which conformations proteins sample and in which order to arrive at such a fast folding process. He just doesn't understand it. Here's an analogy. Let's say you're rolling 100 dice all at once. How many outcomes are there? 6 to the 100th power, which is around 10 to the 77. A crazy huge number. Let's pretend each outcome is like one protein folding pattern. If you roll these dice over and over again, you'll never get the same outcome twice in a trillion years. So how does protein folding work? It's crazy. Okay, now put magnets in one face of every single die and roll them on a metal surface. Look at that. Now you'll get the same singular outcome every time. That's the way the protein folds. And guess what? It's electromagnetism that's responsible in both cases. The conformation that has the lowest energy is the way the protein will fold, just like the magnetic face of each die adhering to the metal surface is the lowest energy for the system of dice. Rob and James can't even grasp this simple thermodynamic concept, let alone the kinetics of protein folding, so let's move on.
Nobody's explained a similar problem, which is how molecules line up together in a cell, how they line up. This is called the Leventhal 2.0 paradox. just in a single yeast cell, just the protein-protein alignment is 10 to the 79 billion. Remember, 10 to the 40th can't happen in the universe. 10 to the 90 is the number of elemental particles in the universe.
It's really more of the exact same stupidity when James says complex molecular ensembles involving the non-covalent interactions of many proteins are so improbable that they could never arise randomly. Using the exact same principles by which proteins fold into a particular shape, proteins also form aggregates due to intermolecular forces which do not require any design, and indeed creating higher order structures consisting of several proteins is easy and is done routinely in the lab. It's just quaternary protein structure. College freshmen know this.
The other thing is, let's say we had them. We can take a cell and we can pull it apart. We can deconstruct it so we can have all the amino acids, or if we can even have the higher order structures, we can have all the polypeptides, all the polysaccharides, all the polynucleotides, the DNA and the RNA. We can have all of that and put each component in separate bottles and give them to any researcher in the world and say, not under a rock, but in your laboratory, could you put Humpty Dumpty back together again? In other words, can you put these components back into a cell? They wouldn't even know where to begin.
This is another one of the dumbest things James has ever said, and he's said it many times before. Of course we can't assemble a cell piece by piece like it's a Lego set. Guess what? Nature never did that either. The first living system was already the product of a long line of evolving systems of molecules. This is just like his conversation with the idiot Stephen Meyer and the Humpty Dumpty problem.
Well, it's the Humpty Dumpty problem, right? You've got all of these pieces, but all those pieces does not a living organism make.
And here's Rob insisting that nature somehow opposes the formation of life.
It seems that nature is telling us that natural processes oppose the formation of life through step after step that you went through.
He clearly never read all the papers I provided about systems dissipating free energy and the formation of life being the direct consequence of the second law of thermodynamics. And again, James was just daydreaming about Jesus when 3 people were discussing exactly this at the Harvard Roundtable, where James pretends nobody was discussing anything relevant to the topic but him.
Thermodynamics actually favors this because everybody talks about equilibrium thermodynamics. That's the theory we're all taught, and not about Prigozhin's work and non-equilibrium thermodynamics, which says simply that if you have enough species to start with, chemical species, you're sufficiently far from equilibrium, which describes pretty much all of the universe that we know, and you continue to import free energy, like this planet, or then it is inevitable that ever more complicated regimes evolve. And this is mathematics. It's been proven, and it's been verified at the simple chemical level with various kinds of reactions that simply do that, set up that experiment. So that to me says life is inevitable. Just wait long enough, because we started.
Why England was doing. That's what Freeland in my paper did explicitly. The thermodynamics of it, favors metastable states, not the stability that you guys are looking for, but metastable states that have a differing lifetime. And this is key. So it's all selection.
Well, nothing lasts ultimately.
Yeah,
and the same is true for any individual life or for life itself.
And it goes way back before the cell ever became.
James, you're like a lazy undergrad who falls asleep during every lecture and then complains that what's on the exam wasn't covered in lecture. Anyway, wrapping up this video, prompted by Rob, James reveals what his next move is going to be. He's going after the textbook manufacturers.
Let's deal with the primordial soup model. This model has come out not by you current-day origin of life folks. This model actually came from the Babylonians. And out of that primordial soup also came their gods. So life came out of that primordial soup, and their gods came out of that primordial soup. It's a stupid, idiotic model that's thousands of years old. That's about 3,000 years old. And then people have run with this thing. And this is what everybody is taught in school. So let's first start by getting that out of the textbooks. And I think they should just take it all out and say we're clueless.
Yes.
We have no idea how it happened, and everything that we've tried, it hasn't worked. So we have no hypothesis on how life originated, number one. Number two, we have never been able to even make life in a laboratory, nowhere close, not even a single bacterium, nowhere close to making life in a laboratory. And just say it, just like that. And let students go away and say, hey, this is an interesting problem that maybe we want to work on. But stop this nonsense. I mean, we can take one of these chapters and compress it to one paragraph. That's it. Clueless. That's what I think should happen.
Yes, James is whining about the Babylonians again, so I don't think he caught my previous comment about his Hebrew mythology getting lots of ideas from them. And he states his goal: get real science out of textbooks and put in some religion instead. Yes, that's right. The primordial soup model, which just says that a mixture of molecules formed the first living systems, should be replaced with "we are clueless." Can you imagine a biology textbook doing this? How about all the chapters on evolutionary biology? It's just a theory, and theories are dumb. We are clueless. Let's keep going until the textbook is the Bible. This really is astounding to hear, even from the mouth of such a lying fraud. 70 years of research, the evolving field of systems chemistry, which again is what primordial soup would really be called if the people who came up with that phrase 100 years ago could have known what we would be researching today. None of it is real because James says so. James is clueless, so everyone else must be too, because he's the smartest boy in class. Allow James to remind us that beyond ignoring 70 years of science, he has no clue what primordial soup even means.
And of course, there are different models. It's not just prebiotic soup. It could be a hydrothermal vent or whatever it is they have in there.
Yes, smelly pond.
Primordial soup, hydrothermal vent, smelly pond. He thinks smelly pond is different from primordial soup. James, what is the smelly pond model? Go ahead and explain that to me and how it differs from the primordial soup model. And while we're at it, what is the primordial soup model? Are you sure it's the lightning lizard, even though I humiliated you in the debate about this exact lie and you just ran to the board and played with chalk?
Why would you tell this lie?
They talk about the primordial soup model in all of these textbooks. That's your question.
This is the primordial soup model. It just means some molecules in water. That's what it means.
Look, you were absolutely clueless on polypeptides. You never gave me the coupling for that reaction over and over again.
Turning into slithering creatures is in textbooks. Show me the textbook. Show me the textbook that says molecules form into slithering creatures.
Because this is exactly the model that the molecules come together. These form higher organisms that come out of the water.
Molecules form higher organisms that come out of the water.
Yes.
Show me the textbook that shows
I don't
that.
have the textbook. I showed you. There they are. There they are. The smelly soup model. There, you have the list right there.
We're all still waiting for you to show us any textbook in the world that says what you're pretending they say, James. Can you really take a whole chapter and compress it to one paragraph or one word? I tried to show you what these textbook chapters actually look like since you've never read one. Do you care to try and compress this into a single word? Show us, James. Oh wait, you'd have to actually learn about it first, and that would demolish your entire scam. This is why I will never stop exposing and humiliating James and the rest of the DI. These bozos are really serious about this. They are going to take all the millions of dollars from their wealthy Christian nationalist donors and actually try to change science textbooks, just like they went to court and tried to put religion in science classes. They will use all their money and resources and political influence to assert their Stone Age beliefs on public life, in clear violation of the separation of church and state. They are actively trying to poison society and bring us back to the Dark Ages. There is no amount of humiliation I can throw at James that will ever amount to what he deserves for this despicable behavior.
Well, thank you so much. And let me just say why there's not a whole lot of other people coming forward. It's because of what the community does to the careers of people who are nonconformists. You take on something like this, and the community will blacklist you in many ways.
Poor baby, he's been blacklisted. No, James, everyone just knows what a lunatic you are and wants nothing to do with you. Remember when you admitted exactly this?
I've been ostracized by not just the Origin of Life community. I've been ostracized by by much broader than that. I go into meetings sometimes and people will kind of look and they'll get hushed. I go into gatherings and like, Tour is here.
You did this to yourself, and everyone knows you're funded by the DI, so you're financially obligated to lie for them. This is your crusade, and you'll follow it until someone stops you by force.
Origin of life research is a scam. We're going to put up here the quote from Lee Cronin because he says I didn't say that he said it was tongue-in-cheek. On his original tweet, Lee Cronin did not say that this was tongue-in-cheek when he said origin of life research was a scam. He said that a year later on Lex Fridman's podcast.
Remember when I bet that James would keep using this tweet even after Lee called him out for misrepresenting him right to his face? What did Lee say he was going to do if James didn't stop?
I clarified that with Jim a number of times, and he keeps saying it. And there's going to be a point at which I have to just get a cease and desist and say, if you keep purposefully making stuff up, I am going to basically suggest with the legal means that you stop that.
Man, wouldn't it be funny if Lee sued him? I do hope James will keep playing with fire like this. Anyway, that's about it for this video. Again, the title says they're going to talk about RNA replication, and they basically didn't at all. Rob brings up one Joyce paper, spins it into failure for the whole field, and ignores thousands of other much more relevant papers, just like every creationist has always done and will always do, because they want to misrepresent the field as being very limited, and also because they have no idea how to discuss this science. The entire video is just James spewing his greatest hits that have been debunked 1,000 times. Luckily, with all the attention James is attracting, there's plenty more to say regardless. Remember when we heard from Andrew Barron, Jim's former colleague? He's the one who showed us that James is the king of hype and commits plagiarism on the regular. Well, guess what? Another one of Jim's former colleagues, Kevin Osman, was emboldened by Andrew's initiative, and he reached out to me with some stories about James that are even more pathetic than the ones Andrew told. Want to hear them? I know you do, so here we go.
So thanks so much for reaching out. It sounds like you have some interesting stories about James. Why don't you just go ahead and let it rip, I guess.
Sure. Let me start by sort of explaining why I'm doing this in the first place.
Okay.
So I have a lot of ties to Rice University. I got both my bachelor's and my PhD from the chemistry department there. I was a postdoctoral researcher there under Nobel Prize winner Richard Smalley for a year. And then I was executive director of a National Science Foundation funded research center at Rice for 5 years. I interacted with Jim both when I was a postdoc and when I was affiliated with the research center. I've been following your video back and forth with Jim since you started it, but upon hearing the comments from Andy Barron in your most recent video, I realized that I could corroborate or even expand on some of the stories you mentioned but didn't go into detail yet. And so I wanted to see about doing that. It's an interesting question, though, why I didn't speak up about this sort of thing earlier. Because it has been quite a few years since I've been at Rice. And the reason for that really says something about the sociological dynamic here. Jim is, or rather was, a powerful individual. Crossing him was considered by many to be a career-ending move because he would take it personally and he would use his connections vindictively. So from a career standpoint, it just wasn't smart to speak out against him. But three things have changed now. The first is Jim is finally losing his influence. The way he was treated at the Harvard Faculty Roundtable you did a video on really shows that pretty clearly. Secondly, I've recently left academia for industry, and I'm quite secure in my career, so Jim isn't really a threat to me anymore. And third, we're in a political climate now where standing up for the truth in our lives has become dramatically more important than it used to be. And as ever the idealist, I feel like I want to follow that ideal. So you'll see the connection of this, the Jim's habits of being vindictive and abusing his power in one of the stories I'm going to tell you. So the issue is, for me, Jim is an embarrassment for Rice University. It's frustrating to me that he is tarnishing Rice's name by spouting his unscientific nonsense and calling it science. What I want to do here in this conversation is to add my voice to the growing chorus that says, no, Jim does not represent the Rice University that I know and love. So that's my motivation. I have 3 stories about him that I wanted to share that bear on different aspects of that. None of them are about abiogenesis, which I know is, what spawned this whole thing. But they do speak to the connection for Jim between his religion and his character, how he approaches science, and his character as a person.
Mm-hmm.
So,
great. Thanks very much for agreeing to do it. Yeah.
Yeah, happy to do so. So the first story is when I was a postdoc in Rick Smalley's lab. I'm a co-author on a paper with Jim Tour. We were working on ways to solubilize single-walled carbon nanotubes in water. And so what we were doing was wrapping them with amphiphilic polymers to isolate the hydrophobic material, the nanotube, from its hydrophilic environment, the water surroundings. The paper that was published on this effort had Jim as a co-author. But his entire contribution to that paper was to make some suggestions to Rick that Rick's students had already made. There was nothing that Jim had suggested that was in any way new. Now, I know this because I'm the person that wrote this paper. When I was working on the author list, I was told to include Jim because it was politically the right thing to do, and that he was aggressively persistent and vindictive enough that we were better off just putting his name on the paper. Now, according to Google Scholar, that paper has over 2,300 citations, which if you look at Jim's publications list, that would put it about 5th in terms of most cited papers that he has. And yet he contributed nothing to that paper. And it makes me wonder how many of Jim's 700 plus publications are like that, where he wormed his way onto a paper without ever having actually contributed anything meaningful to it.
Right. Yeah. This is not his own grad students that he didn't do any work. This is a completely unrelated group that he
That's
just
right.
had absolutely nothing to do with it whatsoever.
Exactly correct. He had like a hallway conversation with Rick at one point. I talked to Rick about what Jim had said in that conversation. And I pointed out to Rick that, all of those things that Jim said to you, your 2 main students on this paper had already pointed out to you, and Rick agreed with me on that. So it's like, so frustrating, very frustrating. My second story about Jim. This was when I was executive director of that research center at Rice. One of the center activities that I helped manage was the educational outreach portfolio. We had museum exhibits, we had teacher education programs that were run by pedagogy professionals from the education department. We had a research experience for undergraduates, we had continuing education programs. It was a very, very broad portfolio of programs that we were overseeing as part of this center at Rice. Jim had this pet project, however, that he felt should constitute the entirety of our center's educational outreach activities. And purportedly, he tried to pressure the National Science Foundation, our funding agency, to make us take that program on. It's called NanoKids. And so what was this, miraculous thing Jim had done that should replace our entire educational portfolio? Well, Jim had drawn skeletal structures of organic compounds that looked like stick figures, the crayon drawings of people with sticks for the bodies, arms, and legs.
Mm-hmm.
Jim then had his students synthesize those things. Jim envisioned these stick figures as being characters in videos, games, and other educational materials who would then explain science to kids. That's it. That was the entirety of his idea, the whole fundamentally flawed idea.
As
By
though,
flaw,
making a stick figure molecule somehow teaches anyone anything about chemistry. Yeah.
right. Well, from a pedagogical standpoint, who's the audience? The stick figure characters would be appealing to elementary school students, perhaps, maybe junior high students, though even that's a stretch, but nothing past that. Who is going to understand what these skeletal structures are? Typically,
Yeah.
what's that?
College students would, but
Yeah,
that's about it.
sophomore level organic chemistry students. I mean, maybe the occasional advanced high school student would have seen them, but for the most part, college students. Just a fundamental disconnect that makes the entire idea not just unworthy of funding, but laughable. I mean, fortunately, we were able to get away with just giving Jim some token support. But this story, to me, illustrates the general lack of perspective that Jim seems to suffer from. You know, he has this idea, he thinks it's the best thing since sliced bread, and so everybody's funding should pour 100% of their money into it. It's narcissistic, is what it is. Okay, my third story is the main one that I really want to talk about, though, because it was so personally offensive to me. While I was executive director of this center, I still interacted with my former postdoc advisor, Rick Smalley, quite a bit. He started out as the faculty director of the center, and then once that position was handed over to another faculty member, he continued to be a key contributor and a driver of the center. Unfortunately, in 2005, Rick died from non-Hodgkin's lymphoma. His last year or so, and especially his last few months, showed a decline in his mental acuity. It saddens me horribly to say that because he taught me so much about how to think as a scientist. During that period, Jim dug his claws into Rick mercilessly. After Rick was dead, those conversations, he dug his claws into Rick by things like private conversations at the hospital during Rick's chemotherapy treatments. After Rick was dead, those conversations were described very publicly by Jim as a conversion to Christianity. Rick's funeral was among the most disconcerting events I've ever attended. Jim had taken control of the entire event, and of all people, Hugh Ross was the best I can describe it is as the keynote speaker, as though it were some kind of faux scientific conference. I sat in attendance accompanied by multiple faculty that had known me extremely well for over a decade and who'd worked alongside Rick for even longer than that. Ross gave a speech that lauded Rick's conversion, seeing the light, as it were, and then used the platform to promulgate the idiocy of intelligent design. The comments I heard from my colleagues aren't fit to repeat in a public forum, so I'm not going to. Since then, I've had numerous examples of interacting with former students and postdocs of Rick's that were unable to attend the funeral. I've described to them the last year or so of Rick's life and this experience at the funeral, and I've uniformly received the same response: horrified, incredulous shock. The picture that Jim has so publicly spread about Rick Smalley in the wake of his death was utterly unrecognizable to them, just as it was unrecognizable to me. And I'd been interacting with Rick within weeks of his ultimate demise. Now, is it possible that Jim, through his long hours of milking his relationship with Rick during Rick's waning hours, might have greater insight into his ultimate character than I do? Yeah, it's possible. I don't know how likely that is. To me, Rick was a giant. He had flaws, but ultimately, in every interaction I had with him, he pushed the edges of our assumptions toward a greater understanding. He was never satisfied with theory when experiment was available. And when experiments were possible, he pushed us hard to do those experiments. He was occasionally wrong, and I can cite several examples of that. But when he was proven wrong, he took the evidence immediately and adjusted his position. And then he began speculating, open to public criticism from everyone around him, what this new understanding meant for what we were trying to do and about the reality we were living in. What bothers me so much and makes me just livid is that Rick's last year or two were hijacked by Jim. And in the weakness of being close to his inevitable death, Rick didn't sufficiently safeguard his legacy against being appropriated by someone who was so obviously agenda-driven rather than data-driven, as Rick was himself. Now, my personal impressions of Rick, while it matches those of his other students and postdocs in my conversations with them, that doesn't prove that that Jim was misrepresenting what happened in those conversations with Rick at the end of Rick's life. But I really only see three possibilities for what could be going on here. Either Jim misrepresented Rick's positions, and after all, the only other witness to those conversations is now dead. Or two, Jim fooled himself into believing that Rick had actually converted, and wishful thinking can go a long way towards making you hear what you want to hear. Or Jim preyed on a respected colleague in decline, successfully converted him, and then crowed about that conversion afterward very publicly for his own purposes. So if you look at those three options, either Jim is lying, he's stupid, or he's reprehensible.
Yeah, or a nice cocktail of the three.
Right. I mean, it's...
To me, it's very clear. He wanted to announce that a Nobel Prize winning scientist was a Christian. So he fabricated the story and then used his funeral as an opportunity to evangelize. That's basically it.
I mean, I am not willing to say 100% that it is fabricated. And one of the reasons for that is that there are quotes from Rick during the last couple of years of his life that you can find that where he is expressing Christian beliefs. That supports the possibility that Jim may have to some extent converted Rick.
Still, though, I think because Andrew told me this story as well, and even if any of that's true, to hijack a man's funeral, to basically do it was like he's selling a book. This guy Hugh Ross, he's basically just selling a book, and they didn't even talk about the guy's chemistry. He won a Nobel Prize. And they talked apparently for about 20 seconds about his chemistry. His son ended up talking about his chemistry. It's like the level of disrespect is off the charts with this. It is. Yeah.
And it is. It has left Rick's legacy just nowhere near what it should be, given the giant of a scientist that Rick was.
Mm-hmm.
And it frustrates me to no end.
Well, I'm just glad that between you, Andrew, and then we'll see who else has something to say, that this truth is finally coming out. As you say, I'm not aware of Jim's influence and like how strong it has been in the past and whether or not it is waning today, but it's about time. It's about time the world sees what James really is.
Yeah. Unfortunately, I don't know how much can be done about it. I mean, there's the whole tenure system that's in place. but eventually bad press is may catch up.
It might. Yeah. I'll
I
settle
don't
for
know.
bullying him on the internet, but wherever that leads, we shall see.
Wow, that was pretty rough. But hey, do you want some more? Because I've got more. Here's an email from a researcher who works in Jim's bread and butter field of graphene manufacturing. They want to remain anonymous since James is such a vindictive bully, but they said I could share the email. They tell me about a paper James published in Nature in 2020 about flash graphene. Surprise, surprise, it's garbage. There is essentially no evidence they even produced graphene. The spectral analysis shows it's really just graphite and tiny amounts of graphene that are no more than what can be found in a pencil mark, since graphene is just single sheets of graphite. They go on to explain what they think James really did and point out how utterly insignificant it is. And look at that, this one fraudulent study was spun into a whole series of further reports in prestigious journals for one long pathetic hype parade. Because why, James?
Flash graphene changing the world.
That's right, Jimbo. The king of hype strikes again. If you insist that this one researcher must have no idea what they're talking about, the cat has gotten out of the bag elsewhere too, and any place this paper is being discussed, on Reddit or YouTube, there are plenty of people commenting who clearly know what they are talking about and agree that it's total bullshit, and they all agree on the specifics of why it is bullshit. Some of these comments go into great detail and are worth reading in their entirety. Just another demonstration of what a fraud James is, even in his primary niche as a researcher, which makes his H-index and citation count completely fraudulent as well. Anyway, after hearing from both Andrew and Kevin, as well as other scientists who work in Jim's primary field of nanomaterials, a very clear picture has emerged of how pathetic James really is. Take all of the disgusting apologetics out of the picture. We are still looking at multiple accounts of how James lies and cheats to get his publications. Slapping his name on the work of his sweatshop of graduate students, even when he offered them no guidance, or even entirely separate and unrelated research groups, plagiarizing existing work, relentlessly slathering his own work with hype to the point of fraudulence, and any number of other sordid tactics. You have to wonder how many other colleagues will come forward and expose even more of Jim's resume as fraudulent. How much more has to come to light before Rice gives James the boot? And to think, all of this could have been avoided. James said it himself. He wishes he had never gone down this path.
If you could redo your interaction with the Origin of Life community, would you change anything? Yeah. I might not even do it. I'm not sure that it was worth it.
You say that, James, and yet you keep doing it. Not only do you keep up your pathetic apologetics, but you make promises to shove your face into textbook publishing, as though you aren't despised enough by the scientific community already. Do you really need even more negative attention and scrutiny? Is this really what you want?
Is this what you want?
Because as long as you keep this up, I'll keep exposing you, not just debunking your conversations with clueless lightweights like Rob Stadler. I'll keep broadcasting people who come forward to expose your lifetime of shady scientific practices until the whole world knows exactly what you are, even your brainwashed flock. Of course, if you quit this path, you'll lose your Discovery Institute funding, since barely anyone else will touch you at this point. It seems like you're stuck between a rock and a hard place. That must be why you look like you've aged 20 years in the past, too. Remember when I told you several years ago that you could be a scientist or a preacher? You have a choice. You can be a scientist, or you can be a preacher. You can't be both. Take my advice, drop out of science, and just preach full-time. After all, you're not a scientist, you're a scammer. It's time to embrace that and just be yourself. So that's it for this installment on James Tour, the laughingstock of the scientific community. How much more humiliation can he withstand? How much further will he allow himself to be exposed? We'll just have to wait and see. I'll see you next time.